Good day, I am Erica Visantiago from Group 5. Our topic is about immunology. So, first, we differentiate innate from adaptive. So, several factors play a role in the occurrence of infection. One of these factors, or probably the most important of them all, is the defensive powers of the host, wherein each human is equipped with an arsenal of responses that aids him or her in fighting disease-producing organism. The human body's first line of defense helps prevent the entry of organism into our body, while the second line of defense inhibits their growth and multiplication. And finally, organisms that escape the second line of defense are dealt with by the third line of defense, which is the immune response. So the first terminologies are innate immunity, or also known as natural immunity. It is already active form at the time birth and prior to an exposure to an antigen. This is also non-specific, includes barriers that prevents entry of organism. Then, second terminology is the adaptive immunity. This one is the specific activated after exposure to a particular antigen. It is acquired response to an antigen that is initiated by recognition of specific epitopes of the foreign invaders. Another one is the antigen, a substance recognized by the immune system, whether by B or T cells, that serves as the target of the immune response but may not necessarily lead to an immune response. Innate immunity and adaptive immunity. So, line of defense of innate is first and second, while the adaptive immunity is the third immune response. So, physical and chemical barriers of innate immunity is skin, mucosa, antimicrobial subs, while the adaptive immunity is secreted antibodies. So, blood proteins of innate is complement, while adaptive immunity is antibodies. So, innate immunity has phagocytic cells, NK cells, epithelial cells, while adaptive immunity has B lymphocytes and T lymphocytes. Innate immunity has specificity of organisms. Innate has relatively low, while adaptive immunity has high specific antigen. The response time of innate immunity is immediate, while adaptive immunity response time is delayed. Dual nature of the adaptive immune system. Red bone marrow stem cells produce lymphocytes, wherein lymphocytes matured in bone marrow and become B cells. Humoral immunity involves antibodies, which are found in serum and lymph and are produced by B cells. Innate humoral immunity involves cytokines and complement system, while adaptive humoral immunity involves action of antibodies. Lymphocytes that migrate through the thymus become T cells. Cellular immunity involves T cells, and T cells receptors recognize antigens.
Immunogen is any substance capable of inducing immune response, whether humoral or cell-mediated or both. Also, immunogen is a kind of antigen which are not only bind to the B cells and T cells receptor but also mount an immune response. Humoral immunity involves antibodies which are found in serum and lymph and are produced by B cells. Under humoral immunity, there is innate humoral immunity that involves cytokines and complement system, and adaptive humoral immunity that involves the action of antibodies, primarily against extracellular antigens, toxin-induced disease, certain viral infection caused by encapsulated pathogens. Cell-mediated immunity. Components of the cell-mediated immune system include several cell types. These are macrophages, natural killer cells, helper T cells, and cytotoxic T cells. Cell-mediated immunity has four basic functions: provide resistance and aid in recovering from infection to intracellular organism. Important defense against fungi, parasites, and bacteria. Involves in transplant and graft rejection. Main defense against tumor cells.
Good afternoon, everyone. Before giving examples, let me first describe what is primary and secondary lymphoid organs means. Primary, or also known as central lymphoid organs, are the primary sites for differentiation and maturation of the important cells that play an important role in adaptive immunity, which is the T cells and B cells. And one of an example of primary lymphoid organs is thymus and bone marrow. While secondary, also known as peripheral lymphoid organs, are the site of reactivity of lymphoid cells. An example of secondary lymphoid organ are lymph nodes, spleen, and mucosa association lymphoid tissues, also known as MALT.
Different types of hypersensitivity reaction. The first is the immediate hypersensitivity reaction. Involve the immunoglobulin mediated release of the histamine and the other mediators from mast cells and basophils. Examples include anaphylaxis and allergic rhinoconjunctivitis. The second one is the cytotoxic hypersensitivity reaction. Involve the immunoglobulin G or the immunoglobulin M antibodies bound the cell surface antigens with subsequent complement fixation. An example is drug-induced hemolytic anemia. The third one is the immune complex reaction. Involves circulating antigen antibody immune complexes that deposit is postcapillary venules with subsequent complement fixation. An example is serum sickness. The last one is the delayed hypersensitivity reaction or cell-mediated immunity. Are mediated by T cells rather than by antibodies. An example is the contact dermatitis from poison ivy or the nickel allergy. Sorry ma'am, maulan kasi kaya maingay.
We have 5 types of immunoglobulins. First, the IgM. Its structure is pentamer and it has J chain. It is the largest and the earliest antibody that will be found in an immune response and B receptor cells. It is also a predominant antibody in acute infections. Because of its large size, IgM is more sufficient activator of complement. IgM together with IgG can activate complement system. Next is IgG. Its structure is monomer. It is a predominant antibody in the secondary immune response. It is the one that can cross placental membrane. This is how the baby gets the passive immunity from the mother while the baby is in the womb. Next is IgA. Its structure is monomer and it has J chain. It is responsible for the secretions such as tears, saliva, and colostrum, as well as respiratory, gastrointestinal, and genitourinary tract secretions. Next is IgE. Its structure is monomer. It is the one used in allergic responses or parasitic infection. IgE, also called reagenic antibody. The last one is IgD. Its structure is monomer and it has no known antibody function, and its surface is found on many B receptor cells.